Hello everyone, my name is Nate, and today I'll be talking about the work our group is doing using machine learning models to understand errors in human genomic variation. Human genome sequencing has advanced significantly. However, identifying complex variants remains challenging. While you have a general idea of why these complex variants are challenging, for example, variants in repetitive regions, the field could benefit from having data-driven models to predict where in the genome a variant will be difficult to call. We are using explainable boosting machines, or EBMs, to create models for variant calling accuracy that are human understandable. These interpretable models can be useful in understanding sequencing errors during benchmark creation, as well as when comparing variant calling pipelines in different genomic regions. So to explain what an EBM is, it's important to understand the whole context of what a machine learning model could be. At the very simplest, you have linear models, which are just coefficients multiplied by their parameters in the parameter space. That's very easy to understand. And then at the very bottom, we have, at the opposite end, we have full complexity models, which are, it could just be any arbitrary function you want. And it's important to understand and point out here that there's a trade-off between accuracy and transparency. As you get more complicated, you get more accurate, but at the cost of being understood and being understandable by a human being. So for EBMs, essentially what an EBM is, it's a GAM with interaction terms. So we have both univariate and bivariate terms. And each of these terms is very flexible. It's actually a decision tree in our case. But the point is that these models are very, very easy to inspect because we can look at each of these features independently. And importantly, for practical purposes here, the algorithm we're using has a very efficient component to it, which allows us to figure out interaction terms in an efficient manner. Because you can imagine that's a very hard thing to do because you have a lot of combinations to deal with. Also importantly, we did not actually make this model. We're actually using these from a team that developed it at Microsoft, and the repository is at this GitHub link as shown below. So how to actually generate the features that go into these EBMs? In general, the variants we're concerned about come from highly repetitive regions. And by way of example, we can talk about homopolymers. And a homopolymer is essentially just one base pair that's repeated over and over again, maybe with a few differences in between, like in the case of this middle sequence where it's a C between a bunch of Gs. And obviously they're strings, and the matrix that goes into the EBMs understands just numbers. So we have to convert those strings to a number somehow. And the thing we're concerned about with these, it turns out, is the length. So we just convert these to a length, and that's the feature essentially. And this is just one of many features we're looking at. We have a lot of feature classes that all involve the same theme, which is highly repetitive regions. And we're doing the same thing we do essentially above with homopolymers, where we take these regions and find one key characteristic, like a percentage or a length of some kind, and turn those into a feature that goes into a matrix. In this poster, we will be discussing two use cases for EBMs. The first is comparing false positive errors in two different sequencing technologies, here being Illumina PCR Plus or PCR Free. To do this, we use two different VCS from HG002 from either method, which are called using deep variant. Next, we fit two independent models on SNPs or indels using 20% holdup for testing. We included 19 main effects parameters, one of which was a categorical variable representing the PCR plus or free parameter, and 18 interaction parameters where this categorical variable was crossed with all of the features. The output was a binary class consisting of true positive or false positive labels. All features shown here are the sum of three terms: the given feature, the effect of PCR free or PCR plus, and the interaction of the two. The y-axis of all plots is in logistic space, so 0 means that an error is 50% likely, all else being constant. For indels, false positives are more likely in AT homopolymers, greater than 12 base pairs for PCR plus but not for PCR free. The same is true for GC homopolymers, greater than 22 or 38 base pairs for PCR plus and PCR free respectively. For tandem repeats, the trends are less clear, but in general, errors are more likely in PCR plus. For SNPs, the difference between PCR plus and PCR free were much less, but false positives became more likely beyond 15 base pairs in AT homopolymers and at any length in GC homopolymers. For tandem repeats, increasing copy number predicts higher error rates. In general, this illustrates, particularly in the case of homopolymers, how these models can be inspected to find clear delineations beyond which errors are more likely to occur. Our second use case was to assess missed variants, false negatives, in HiFi or Illumina analysis pipelines. Note that in this experiment, we varied both the variant caller and the sequencing technology together, since the former was dependent on the latter. The model training process was analogous to the first use case. However, it used a few less parameters, and a negative class here was false negative instead of false positive, for obvious reasons. It's worth noting also, in the future we plan to use this model to understand the prevalence of misvariants in a ClinVar dataset. The following plots have the same interpretation as the previous use case. For indels, missing the variant is more likely in inti homomers beyond 15 base pairs, and at any length in GC homopolymers. This inflection point is the same for both pipelines, but the overall error rate is higher in HiFi. For SNPs, HiFi is less likely to miss a variant in hard-to-map regions with 2 levels of difficulty, as defined using version 3.0 genome model stratifications. For transposable elements, HiFi in general is less likely to miss a variant, but Lumina shows improved accuracy with calling variants that are in signs versus outside of signs. Finally, HiFi is less likely to miss variants in a segmental duplication overall, and errors are more likely with increasing size and similarity. As in the first use case, these results show clear delineations for where errors are more likely to occur. And I'd like to acknowledge our funding sources, and I'd like to acknowledge you for your attention. Thank you very much.